it is ironic that to do circadian work, you have to disrupt your own circadian rhythm. Once you learn about the circadian rhythm as a biologist, you cannot ignore it. We're studying aging, and we're studying this part of aging called cellular senescence. Cellular senescence is when your cells stop dividing, they stop growing, and they start secreting these inflammatory signals. It's an accumulation of mistimings that can overall lead to damage and changes in gene expression. This can actually be really helpful in some contexts. It's how your wounds heal, it's how your muscles regenerate, it's how fetal development happens within the placenta. But as we age, we can accumulate these cells in our tissues, and they can seed chronic inflammation that can lead to age-related diseases like Alzheimer's disease, heart disease, etc. Is a senescent cell the same at any given time in the day? We are not the same at any given time in the day. So we know we have a circadian rhythm. We know we wake up when the sun is up, we go to sleep when the sun is down, but that rhythm is actually much more complex when we think about it within our bodies. No circadian studies to date that I'm aware of have done full organ studies in genetically diverse mice. These are mice that better model human genetics. So I am not the same as you are not the same as me, but we're working on mice that are all inbred and genetically the same, and so we wanted to get mice that model humans and are all different. The fact that we can get these specialized mice that I didn't even know existed before I came here. At a typical institution, you work with C57 black 6 mice, and maybe you have knockouts on that line, but you don't think about any other kind of mice. Whereas here, you are constantly being introduced to different strains, different backgrounds, different ideas, different types of mice, different ways of doing biology, and it's why I wanted to come here, because there's resources, but people also don't shy away from big challenges. One of the things with working with mice that is often overlooked is that mice are nocturnal. So when we're here during the day and we're phenotyping or sampling, we're sampling them when they're supposed to be asleep. So what we did was we did an experiment every 6 hours for 24 hours. We just had 4 time points, 9 a.m., 3 p.m., 9 p.m., and 3 a.m. And so to do this, we had to get really creative. So we utilized the extra spaces within JAX to use procedure rooms to have mice in the dark where we could easily access them in order to do this testing. Mice can see the light in the hallway. They can see the light, you know, on places where you haven't turned it off necessarily, so they have put up red filters on all of those areas where potential light could be getting in at night. So we used headlamps and other kind of lamp light in order to have enough lighting to see what we were doing, but not enough to affect the study. I regularly speak with my mentors, Ron and Nadia. Nadia is always pushing me to think more about the translation of my work and where we can go next that's really going to help people. Ron is all about, you know, the technicalities of the work. You know, how do we make this work? How is it feasible? What is the best science to be doing? The main motivator for me to just keep going beyond the human, you know, translational ability is the mice. The mice are giving us so much for us to do this work, and I am not going to take that sacrifice in vain. So if that means I have to show up at 3 a.m. and then show back up at 3 p.m., I'm going to do that, because if I can intervene in somebody's health by just changing what time they go to bed and changing what time they eat versus giving them a drug, I think that's what people are more interested in, is having these resources and this knowledge to know how to better care for themselves.